Well, thanks, Paul, for the introduction there. And thanks to the center for inviting me to talk. Yeah, I do send my grants to my brother, but they don't come back. There are a lot of red ink on them, and he would never fund them. So luckily, he's not on any committees at NIH that would review my grants. So, it's fun having him pick apart my stuff. All right, so as Paul said, today's topic will be cytochrome C biogenesis. I'm going to focus on just two things. One is heme trafficking. I'm going to give you an introduction on what cytochrome Cs are. And as Paul said, there is a covalent link. There's two of them in most cytochrome Cs between cysteines and the vinyl groups of heme. And although that reaction is spontaneous, the vinyl groups are very reactive. And if you just bring a thiol group close to it, it will form a thioether bond. And I'm going to show you that with some of the methods or approaches we're using now to study it. But in the C-type cytochromes, of course, nothing is random in the cell, and you have to have systems that do that bring the heme to the apocytochrome C to do the covalent linkage. And I'm going to talk about that too. Yeah. Oh, yeah. Okay. All right. So again, it has thioether bonds, two of them in the crystal structure as presented here. You can see in the yellow one of the thioether bonds to what was a vinyl group. The other thioether is right there. You follow it up, and there's a histidine right here that's in all cytochrome Cs, and that histidine acts as a ligand to the iron of the heme. So one of the very interesting properties I feel that is specific to C-type cytochromes is that the protein doesn't fold until the heme is attached. And so it actually folds around the heme, and the heme is driving that process. For those of you that are interested in heme, it's one of the unique situations where, unlike the globins, the hemoglobins and the B-type cytochromes, those fold as apoproteins, and then the heme inserts spontaneously, usually, into the protein. This is quite different. And I think it's generally accepted in the field now, and it was debatable for quite a while, why you have to do this, or why does life do this. C-type cytochromes have been around for 3 billion years in prokaryotes first, of course. And it's generally agreed now that it provides stability to the protein. Heme is never lost in the protein, particularly in prokaryotes where they're in the periplasm, they're outside. And those are harsh environments at times, and so you have to maintain that heme. And so hence the linkages. I don't need to go over this slide, hopefully too much. The functions of cytochrome Cs are varied. You are familiar with the mitochondrial cytochrome C acting as an electron shuttle in respiration. The C1 of the cytochrome BC1 is also a C-type cytochrome. And so respiration, apoptosis in metazoans is also a function of the cytochrome C. Chloroplasts have the BF. The F is truly is just like a cytochrome BC1 complex, given a different name for some reason, but it has the covalent linkage just like the C1. And then in some chloroplasts, they have a soluble cytochrome C that acts in photosynthesis. In prokaryotes, a large variety of cytochrome Cs. There's hundreds of different types of cytochrome Cs. One is like the mitochondrial and works in aerobic respiration. But there's all these other ones that work in different types of respiration, anaerobic, for detoxification, photosynthetic bacteria require C-type cytochromes for photosynthesis. So a lot of different C-type cytochromes are required by the prokaryotes. And that's probably one of the reasons why the recognition determinant for the prokaryotic systems, and I'm going to talk about those in a minute, only recognize the cis, XX, cis, his, whereas the recognition determinant for the mitochondrial enzyme, the one that Paul works on in Plasmodium, recognizes more than just the cis, XX, cis, his. Okay.
So are all C-type cytochromes, are they requisite that they carry electrons?
In apoptosis function, they do not have to be carrying electrons. It's simply a signal molecule. to the APAF1. And so in all the other cases, yes, as far as I know, they carry electrons. In apoptosis, it has been thought in models that you see that only the iron +3 version of cytochrome C, not the iron +2 reduced version, is the signal. But Deanna Mendez in our lab, with some new cytochrome Cs that she has produced, I won't have time to talk about her work, has collaborated with Chris Akey at Boston to show that both iron +3 and iron +2 cytochrome C is a signal to apoptosis. So that's the only case I know of. All right, this is the reaction that takes place in the C-type cytochromes. As I said, this is a spontaneous thioether linkage that occurs as long as you bring this close to the vinyl group. But in nature, there's 3 systems that have evolved to do that. And I'm going to tell you about the 3 systems just briefly for each one, and what are some of the important properties of each system that does that. So I already mentioned it has to have a reduced thiol and heme. Iron has to be reduced for that reaction to take place. His residue then acts as the ligand. All right, I know this is a very busy slide, a little bit out of focus. You don't need to look at the nomenclature. Just wanted to give you a little bit of history of the study of these 3 pathways, these 3 systems, and how old they are as far as in life and when we started discovering these systems. So we started working when I first got to WashU in 1987. I actually didn't study this my whole career. I actually had an NIH grant for working on nitrogen fixation in photosynthetic bacteria, Rhodobacter capsulatus. And we were studying the signaling mechanisms and how they're controlled by anaerobiosis. But as a lot of science, it led to some mutants, 140 mutants, that had a property they couldn't grow anaerobically. And we characterized those mutants, and many of those mutants turned out to be in the pathway for cytochrome C biogenesis, and we cloned the genes. And that's how I got started in cytochrome C biogenesis. And so in 1989, we published a paper that designated about 6 of these 8 genes were required for making a cytochrome C. And then in 1992, we published a paper in Genes Development that went and published the sequences of most of the genes. And so there's a couple other groups. Hawke-Hennecke worked on not Rhodobacter, but Bradyrhizobium japonicum. That's an alpha proteobacteria, just like Rhodobacter. And Stuart Ferguson worked on Paracoccus in those days. And so we all together discovered the CCM, what we call System 1 now, and the nomenclature is CCM. We didn't even know E. coli had these genes until 1996 when the genome was done. And so that's why E. coli wasn't the system that these were discovered in. E. coli doesn't need cytochrome Cs for aerobic growth. When you grow E. coli in the lab, they don't use cytochrome Cs. They only use them under very special anaerobic growth. All right, so that's 1989. In 1996, there was a protein that we had presented in the G&D paper that was homologous to 2 of ours in what we call a WWD domain. And you're going to hear a lot about that domain. That domain was in this protein called CCSA of chloroplasts, and Sabihar Merchant knocked that gene out, CCSA in chloroplasts, and voila, it didn't make C-type cytochromes. And so that's the first report. Our group worked on Bordetella pertussis for a number of years. Bacteria are always easier to work on than plants or Chlamydomonas. So we chose Bordetella for a lot of genomic reasons and helped characterize these 4 genes. It's a distinct system from System 1. And you're going to hear about CCSBA in more detail as a heme transporter and a synthetase. And the earliest study done in cytochrome C biogenesis was by Mark Dumont and Fred Sherman at Rochester. And they worked in yeast, and they found a single mutant they called cytochrome C heme lyase that could not make cytochrome Cs in yeast. And so that's why it was kind of hard for our papers to get accepted in the first few years. Everybody thought it was going to be just like yeast, and it wasn't. At the end of the talk, I'm going to go over the cytochrome C heme lyase, which is now has a different name, but it's the one that was discovered. All right, a little bit of more history in a technique that we use that I want to go over. We started studying Bordetella in the mid-90s, late 1990s, and characterized the 4 genes as being required in Bordetella pertussis. For a few years, we tried to get this system to work in E. coli. So if you knock out the E. coli genes, all 8 of them, CCM system 1, you put in a cytochrome C4, in this case that's a Bordetella cytochrome C, and it can't put the heme, it can't attach the heme to the polypeptide, cytochrome C4 polypeptide. E. coli grows fine with that because, like I said, it doesn't need aerobic growth for aerobic growth, cytochrome Cs. And it took us a while. We never got anything to work with Bordetella, which are two different genes. CCSB and CCSA are different genes, as they are in Mycobacterium and Bacillus and so on. But we noticed that Helicobacter in the genome sequence has a fused CCSB and A. And a rotation student in my lab, that was his project, to clone this fused gene and try to accomplish something we couldn't do for 2 or 3 years. And within 3 weeks, he had the gene cloned and put in E. coli with this reporter plasmid that had the cytochrome C4, and this is what he got. So pretty nice rotation project. Unfortunately, he didn't join my lab. I don't know why, but anyway, others carried on this work, and Rob Feisner is one of the main ones, and Elaine Frawley you'll see in a second. So this heme stain is traditionally thought to stain on SDS gels only the heme that is covalently attached to a polypeptide. There's some exceptions, and you'll see one. HECS is one that can bind it on the heme. But for the most part, covalently bound heme is the main sort of reaction that takes place here. All right, so then Elaine Frawley carried on with this CCSBA from Helicobacter. And both System 1 and System 2 then in my lab started sort of more biochemistry. And so in the last 10 years, we've been able to purify all the proteins in System 1 and all the proteins in System 2. These are all integral membrane proteins. There's no structure known. They're tough to work with. You get about 0.5 mg per liter at most of these proteins. And so we've dabbled a little bit, but it's really just recently we're getting to the point where maybe some structure can be done. All right, so Elaine Frawley then purified the CCSBA protein and did some tricks with this putative transporter, CCSBA heme transporter. We knew, of course, it was the cytochrome C synthetase because this replaced the function of all of the System 1 genes, but we didn't really know if it transported heme, and that's what her paper was on. And so I'm going to go over some new approaches to study heme transport for the majority of this lecture. And imidazole correction, although maybe not a new method for his substitutions, but for transporters is sort of a new application of an old approach to studying heme transporters. A new method that Molly Sutherland in the lab, a postdoc in the group, has developed, we call it cysteine scanning mutagenesis to trap heme in transporter proteins. Transporter proteins of heme are tough to study. They're not just integral membrane proteins, but if heme is, of course, transiently transported through these trafficking systems. And it's hard to capture the heme in there and to know where it's binding. And so Molly has used the natural tendency of cysteine to covalently link to those vinyl groups of heme to trap the heme in the transporters. And I'm going to show you some of her new data not published on trapping. And then at the end, maybe 10 minutes or so, I'll have time to talk about the mitochondrial system, HECS, that Paul works on in Plasmodium and we work on the human system. All of our work is E. coli, as I said. We get everything to work in E. coli, and then we can do some more biochemistry and so on. Recently, Shalon Babbitt-Ledbetter in the group has, besides working on HCCS function and the structure of the substrates that it binds, she has gotten in vitro reconstitution of cytochrome C biogenesis by a pure HCCS. First time ever, no one's ever done that. And I'll take a few minutes. I won't have time to talk about the Deanna Mendez's work on the new cytochrome Cs. Okay, so let's go back. Let me go back now to System 2. That is the CCSBA protein, integral membrane protein, no structure known. It has two properties that you can guess. It's got something to do with transporting heme. It has two 100% conserved histidines in transmembrane domains, and it has two histidines in external domains. Remember, cytochrome Cs are produced in the periplasm or in the chloroplast outside this chloroplast. And so this kind of got the typical topology and determinants that you would guess a transporter should have. And most transporters that have been discovered, and there's quite a few now in eukaryotes in particular, they have usually a conserved histidine, maybe two sometimes. But it's, like I said, it's tough to study these transporters in vitro and in vivo for many reasons. So our proposal is that heme is liganded by these transmembrane histidines, moves to an external domain called this WWD domain we talked about in 1992 as a putative heme binding domain that presents the vinyl groups to an acceptor protein. Here the acceptor protein is cytochrome C, cis XX cis his. And so one of the approaches that we have used in the last 10 years, started by Elaine Frawley in the lab, is looking at the histidines. And so she and recently Molly Sutherland have done histidine mutagenesis. And here's a function, the histidines, the transmembrane histidines and the periplasmic histidines. This is a heme stain. And you can see that the wild-type CCSBA can put on the heme. There's a little bit of degradation down to a 12 kilodalton protein for this cytochrome C. But none of the mutants in the four histidines are functional. And so to test this in the last few years, we've been using this imidazole correction approach. And Doug Barrick in 1994, I believe, is the first one to do this, and I want to give him a little publicity because I think it's a really sort of unique sort of approach that he started. And he worked on myoglobin in E. coli, overproducing myoglobin in E. coli, and he made a mutant in the histidine, the proximal histidine, that's required for heme binding, been known for a long time. And he calls this the cavity mutant of myoglobin. And when you produce that in E. coli, it doesn't have heme. You purify it as the apoprotein, apomyoglobin. And so this is the figure I really think is elegant that I try to publicize quite a bit. He took whole E. coli cells and added to the growing E. coli some imidazole, right? From 0, which is right here, to 10 millimolar imidazole. And he can do a carbon monoxide difference spectra, that's what this is, and you can see in growing E. coli how much heme, how much myoglobin that has heme is made. And so with adding increasing amounts of imidazole to the culture, imidazole gets into the E. coli and it ligands, it's hydrogen bonded of course by other side chains, but it acts as the ligand to the iron of the heme, and that is enough binding energy for the heme to bind to that site. And so from 1 to 10 millimolar of imidazole was functional, and Roach et al. Did the crystal structure to show that imidazole is in that cavity or that pocket. And so we've been using that approach since we've been able to get function of a lot of these proteins in E. coli. And so that's what first Elaine and more recently Molly has done with the CCSBA and some of these other trafficking proteins is the imidazole correction on heme transporters. And so imidazole correction is shown here. This is Elaine's work. For the transmembrane histidines, this is E. coli growing anaerobically, E. coli growing aerobically. Here's the wild type. You see plenty of cytochrome C4. Right there, when it grows anaerobically, you get function of the CCSBA histidine mutants by adding the imidazole. Imidazole goes into the pocket or the cavity that is formed in CCSBA and corrects for the function. Surprisingly, the function is considerably less aerobically than anaerobically, even though the wild type, it's the reverse. And so Sabia Merchant wrote a commentary on this PNAS paper. I think the title of the commentary was His Protects Heme. And so the point is that not only is this a heme binding site, the transmembrane histidines forming a heme binding site, but the function of those histidines is to protect the heme from oxidation, right? Because only iron plus 2 can be ligated, that is covalently attached. And so I think that was a perfect sort of use of an old approach for transporters. Wei-Fen Zhu, a past postdoc in the lab, extended this further, and she made mutants, not just alanine, but glycine, in the transmembrane His, and the two, the red and the yellow, are 1 and 10 millimolar imidazole. There's no function without those. And tyrosine, when you change the histidine to a tyrosine, it doesn't correct. We were originally thinking maybe tyrosine can be a ligand also. There's some very interesting redox properties of tyrosine that we were interested in. But it did not function with a tyrosine, nor could it be corrected by imidazole with a tyrosine, which that makes sense because we don't have a cavity anymore, right? The tyrosine side chain fills up the cavity. So you can't get imidazole in there. All right, so the next question is heme trafficking through this WWD domain that was proposed in our 1992 paper as the heme binding motif. This is a superfamily of proteins. Milton Sayre at San Diego keeps the database of transport proteins, and he calls this superfamily the putative heme handling proteins because of some of our literature that we propose it is. But no one has really proven that heme traffics through this WWD domain and presents the vinyls, and that's what Molly Sutherland in our group has been working on in the last 2 years. So in system 1, now we're switching over to system 1, and system 1 has these WWD proteins too. That's where we first discovered them. One of the WWD domains is in CCMC, so the proposal is that heme moves through CCMC, transmembrane regions, and is covalently linked to CCME. That's what this little black bar means. There's a covalent linkage to its acceptor protein, CCME. That's been worked on a lot by Stuart Ferguson and Linda Thoney-Meyer. And so we haven't done the majority of work on CCME, they have. But we've worked on the complex, this putative transporter complex of heme. And so it has a WWD domain. Then the heme from CCME is shuttled over to the synthetase, and we've done a lot of work there, and many of these papers have to do with this cytochrome C synthetase, where there's another WWD domain that presents it to its acceptor, the apocytochrome C. So the next 10 minutes or so, I'm going to focus in on the WWD domain and how we tackled this domain to show it truly is a heme binding domain. Okay? So CCS or CCM ABC is right here, diagrammed right here. Feisner and Cindy Richard Fogel in the group has worked on this particular complex quite a bit. If you mutate CCMA, which is an ABC transporter, or B, and you can even mutate a single site in A that doesn't allow this hydrolysis, you end up with a complex that has CCME and heme, the heme here in red, as a tight complex, okay? And so a lot of results suggest that the function of this ABC transporter complex is to release the heme that's still bound in C, to release it as the chaperone, the heme chaperone here, with this covalent link to the histidine of the CCME. And Cindy in our lab deleted A and B, and she is able to purify a very stable CDE complex. D is a polypeptide of 62 amino acids. You don't see it on the purified complex, but we know it's there. And we call this the post-addict, meaning it's after the covalent link to the His 130 is made, intermediate complex, because it's on the path for the trafficking of heme. And so we've studied this complex in the last few years, Molly Sutherland in particular, to analyze the complex. So here is just a cartoon of CCMC, 6 transmembrane domains. Here's the WWD. Here's 2 histidines we know are required. And here's CCME, a single transmembrane domain, and a covalent link to histidine 130 right here to the vinyl 2 group, work done by Linda Thony-Meyer and Stuart Ferguson. The only structure known for the whole system is the apoprotein of CCME when it's truncated here. So the apoprotein structure is known, and you're going to see that in a minute. So we'll get back to that apo structure in a second. Our goal is to prove, one, that heme is binding to WWD, and two, where is it binding in this complex, and in particular to CCME where the apo structure is known. So Cindy started this and Molly has continued it. We get a very nice pure complex of C, a little bit of GST star because it's a GST functional GST CCM fusion. And then we get CCME. The heme is on CCME. You can see it's very large amount of heme that is covalently linked through that His 130. And I usually promise the audiences I won't show spectra, but you guys are used to seeing spectra possibly. But there's a very unique alpha in the reduced form of this complex with a split alpha that's diagnostic for this heme with the one covalent link and the complex. All right, so there's Molly Sutherland, who has done this work. So bottom line is she's made, I think, 70 to 80 mutations where she takes cysteines and substitutes in cysteines to try to capture covalent links to the vinyl groups of heme to sort of map out where heme is binding as it goes through, right, this transporter, and ends up in this tight complex that it doesn't have CCM A and B there to release it. So that's how we're able to do this, a little genetic trick. And so hypothesis, WWD domain binds heme and presents the vinyl groups to an acceptor protein. The acceptor protein in the case of C is ApoE. The acceptor protein for CCSBA is the apocytochrome C. So first we started with CCME. That's the one where the apo CCME structure is known. And we peppered it with 16 cysteine substitutions. They weren't just random. You'll see why in a minute when you see the structure. We really honed in on where heme might be binding in the structure. And so the wild type CDE complex has the heme in the holo CCME. That's the same thing I showed right here, right there. If you mutate the histidine right here, all of the heme is still stuck there, right, in the complex, but the heme is free B heme, right? So it's a B-type heme. When you run it on an SDS gel, it runs at the die front, okay? And so in the cysteine scanning mutagenesis, we did it in that background because we're looking for something that covalently links to CCME now with a vinyl group that we've put in a cysteine residue, and so we're trapping the heme in there. And so I apologize, this is a busy slide. We're getting it ready for publication. And so I'm just going to point out a few points of this slide. This is the structure of the apo-CCME. No one has been able, and this was structures by Enquist et al. In 2002, and MR. As I said, no one's been able to get the structure of any of the proteins except this one in System 1. And there's no heme in it because that's technically a tough experiment to get heme in it and do the NMR structure of that. But we wanted to find out or to determine where the heme was binding in this apo-CCME in our complex. And so what we show here is this cysteine thiol in this orientation, okay, is located in what we call the vinyl 2 pocket, which is where His 130 would be if it wasn't mutated in here, right there. And that vinyl 2 covalently links in the wild type to that histidine right there by a typical sort of histidine vinyl crosslink. And so we mutate that, and we do the saturation mutagenesis of all of the residues here and here, and one gives us this covalent link. It's next to the His 130, and we have proof that this is binding to the vinyl 2 also, this here. And it allows us to model in heme into the complex with the vinyl 2 group here and the vinyl 4 group in this pocket. Unfortunately, this pocket many of the residues, the completely conserved residues are in red, are required for the folding of CCME. And so you can't trap heme there, but we have some other methods we've used to prove that that is a vinyl 4 pocket. And so heme theoretically goes through CCMC, and in this WWD domain, just cartooned here, is presenting the vinyl groups to the E in the complex. And so Molly did saturation mutagenesis of the WWD domain to try to capture covalent links between these. And I'm just showing crude data here that she got some mutants, right? And this GST CCMC has a little bit of the free B heme that stays on an SDS gel. And so that's sort of common for some heme proteins that aren't covalent links. You do get a little bit. Most of it's the free B heme, but if you overexpose it, you can see a little there. Certain mutants have a lot of heme proven to be covalently attached in various cysteine mutants like this W114 cis.
So,
yeah.
Can you go back one?
Yeah. Yeah, sure.
So the propionates that are still there on C, do they interact? Do you know where they interact?
No. The WWD domain is up to 20 amino acids. We presume that they are interacting with residues in the WWD domain, and that it's got to be very spatially rigid to do this. This is all new data, and now that we know where the crosslinks are making and where the vinyl groups are in the domain, we're going to start doing some modeling with that domain. But it hasn't been feasible until now, but we just haven't got to that point of doing some modeling. Okay, so there was 3 that were particularly interesting covalent crosslinks, and I think it was 5 total, but these were the major ones in the WWD domain that cross-linked. And so initial results then indicate that there are 2 or 3 covalent links, one to this W114, this is the WWD domain in CCMC, and then the 2 here. Okay, so the big question being which vinyl group is attaching to which of the different residues. Giving an example here, we now know it's the case, that this residue covalently links to the 4 vinyl, and these 2 to the 2 vinyl. That's a proposal. And then Molly and Josh Jardowski, technician in the lab, have done some studies to confirm that. And so what they did was a double cross-link. Remember, the His-130 in our original experiments was mutated to an A so we could detect the cross-links in CCMC. And so what they did was make the mutations in a H130 background, so we definitely will get the 2 vinyl cross-link here. And so if this is the case where the W is covalently linking to the 4 vinyl, we should get a species that gives us C and E on an SDS gel. You guys are going to think that the only thing my lab does is heme staining, but we do other things more than heme staining, but we use it as a major technique. We even have a couple patents on in vivo heme staining, but they've never made any money. But anyway, this property is predicted if the 4 vinyl is and the 2 vinyl to the H130 is occurring. And likewise, if these are occurring at the 2 vinyl, likely this will be the major species because that's the one that works in nature. And that's exactly what happened. If you look at a Kamasi gel of these mutants, these are the cysteine changes in the wild type background. We get a species that's the molecular weight of CCMC, GST, CCMC, and E right there. It heme stains. So we have basically 3 species in this mixture, the purified mixture of this membrane protein complex. One is CCME that has this cross-link. One has CCMC that has just this cross-link, and one has the full cross-link here. The other 2 only form this cross-link as expected. And so just to prove that that has CCME, and then a GST blot also shows it has the GST CCMC. All right, so they've shown that this is the species This is what is happening, and the W114 is covalent linking to the 4-vinyl, and this one to the 2-vinyl. We have pyridine, hemochrome spectra, and trypsin, digest, and so on to confirm these heme stains, but I'm not going to show you those. And then she peppered the WWD domain of CCSBA. That's the one right here. The acceptor is apocytochrome C. Here's the WWD domain of CCSBA. So they mutated all of these residues, and each one numbered covalent links to a certain extent. There's the data. These are the ones that form the crosslinks, the covalent crosslinks in the WWD domain. These two are the same positions and ones as the W and the R of the CCMC. And when we got this result, we were just sort of blown away because we really have mapped the 4-vinyl and the 2-vinyl now in the WWD domain in two different subfamilies of the of this superfamily. And so anyway, this particular protein, CCSBA, in the WWD domain that's there, is a transient. Remember, in CCMC, we captured the heme there using a genetic trick, right, in the complex. In CCSBA, it's transiently moving through. And so this, to me, shows that we've proven that we can trap heme with cysteines even in something that's transiently moving through. It's rapid enough. You can guess based on chemistry and so on that it might happen, but I think this proves that it does happen. And this is just her gels of that. And so the conclusion is the WWD domain is truly a heme-binding domain. We can now tell Milton Sayre to take off the putative, I believe, in his heme-handling proteins, HHPs, because that's its function. And this is just the three subfamilies. The CCMF is the synthetase of system 1. We obviously now can do the same experiments with that. All right, the last 5-10 minutes here, I'm going to go over our mitochondrial system that we've been working on just for the last 5 or 6 years. Quickly, I'll go over some published data already. The midazole correction of the HECS histidine that is involved in binding the heme. A little bit about purification. And then I want to end up with Shallon's newest data on the in vitro. So Shallon Babbitt-Ledbetter is the one she's been a postdoc in the lab for approximately 4 years now, and has been a great colleague and researcher. This is HCCS, identified 25 years ago by Dumont. Now it's called HCCS instead of CCHL, just simply because the human genomics people call it that, and so everybody now refers to it as HCCS. It's responsible for a disease called microphthalmia with linear skin lesions. It's an X-linked, so it's only in females, this disease. It's lethal in males. Shallon has a paper in biochemistry that shows that this mutation, one of the major ones of this disease, is in heme binding. And we have quite a bit of data on this E159 residue, not just the K, but an A substitution and so on, and they're involved in heme binding. And she can correct that in E. coli with just adding ALA to the culture to increase the heme levels. So heme is always important in this particular enzyme. And this is a quote by James Allen in a review he wrote in System 3. This is System 3. Lack of a structure, expression system, and so on. And that led to our sort of trying to work on this. And Brian San Francisco in the group is the one that first worked on this in our lab. And he purified the protein, showed that His154 is necessary for binding the heme. That's the first step. Step 2 is after the heme binds, it is recognized by cytochrome, it recognizes cytochrome C. Heme is absolutely essential for that. Shallon has a JBC paper that shows the alpha helix 1 is important in that recognition. Shallon also has a paper that shows the histidine 19 acts as the second ligand in this complex that can be purified. And then, and only then, is the thioether when the his19 ligands and the alpha helix 1 binds is the cysteine, in this case, this cysteine and this cysteine thioether linked. And then it is released. Step 4 is the release of the cytochrome C from the HCCS. Resonance Raman of this complex shows that it's distorted. We believe that distortion is part of the reason that it's released from the heme in here. There's a natural distortion or ruffling in C-type cytochromes. It's present in all C-type cytochromes. All right, so we can purify HCCS. It has some heme. That's what I was telling you. It's not covalent, but it sticks there when you don't boil the sample. It does remain in the sample. Some spectra. The H154, it doesn't purify with heme. It doesn't have heme. Same thing, imidazole correction is occurring with that particular His 154. So histidine 154 is the key binding residue. Here's Shallen's alpha helix 1. This is just a model. Alpha helix 1, particularly the Phi 11, is important for positioning the cis 15 to get this thioether linkage. We can purify the complex, shown here. H154 does not purify as the complex, therefore heme is absolutely necessary to bind the apocytochrome C. And let me switch over. This is published in Shallen's JBC paper. And I'm not going to go over that. Let me just give a shout out to this before I tell you about our in vitro stuff, which I can end with. Shallen did this work with Deanna as far as this particular figure. Shallen changed all of the histidine 19, which is in the conserved cis XX cis his motif, to a number of different residues, and then determined whether you can make a cytochrome C. And they did a lot more work than this. They purified the complexes to see if the methionine could act as a ligand and so on, spectrally and so on. And only methionine, H19 methionine, could actually act as a ligand. Methionine is the second best ligand to iron of heme. Histidine is the best as far as strength of the bond. And so only methionine could substitute. It substituted at a very low level, right, in using HCCS wild type, but it still made some what we call the bis-met cytochrome C. It has a met and in both the His 19 position and its normal met 81 in what cytochrome C normally has. And so Deanna went on, and this is just published last month, I think, or this month, in PNAS, that you can use Shallen's initial HCCS release mutants. They have a tendency to release at an easier rate. They're in the heme binding site of HCCS, these mutations, these substitutions. And E159A, remember that's the MLS residue. E159A is particularly good at making more product because we believe it releases the product at a higher rate, step 4. And so using the release mutant, in particular E159A, Deanna has been able to make enough of these various mutants, this bis-met is just one of them, to study in purified proteins. And so we have some interesting results on the different unique, and a single thioether type that's never been made before with the first cysteine single thioether has also been able to be produced with this method. All right, so question. We're moving now to just unpublished. In the last probably year we've been working on this, and it's been a slow process. Sometimes it needs to be anaerobic, in an anaerobic chamber and done very carefully and so on to get in vitro studies to work. And so now we have it to a point where we can even do it aerobically. So can cytochrome C be assembled by HCCS in a cell-free system? It's purified from the membranes. That's one of the reasons we were successful and others not in purifying this protein. Even though it's predicted not to be a membrane protein, it is a membrane protein in E. coli. It acts kind of like an integral membrane protein. We use Triton X100 or DDM, dodecyl multicide, to purify it. But it's got a heme, and we believe the majority of that is reduced, iron plus 2. Believe it or not, that's been a tough problem in our resonance Raman expert Kent Rogers has done resonance Raman and so on, and believes it is mostly reduced in this form. All right, so then the idea is take this purified protein with the heme, add apocytochrome C. We simply strip the heme off of the horse heart that we buy from Sigma as our substrate, and then see if we can get heme attached and it released from this. In vivo, that's why I put this here. In vivo, you start with this, and you end in the soluble cytoplasm of E. coli, you end with a pure cytochrome C with a 550 alpha peak. 550 right there is diagnostic for cytochrome C. And so this is basically what Shallon and an undergrad, Jen Hu, has been doing in the last year. And so you start with the black, you add them together, and you start to develop the peak at 550 here after 1 hour of incubation at 37 degrees. So we are very happy to see that. It's been proven by pyridine hemochromes. I think it doesn't work with the H154, which you would guess it shouldn't. And you can detect it on a heme stain gel also, the occurrence after 1 hour of this horse heart holocytochrome C. It doesn't happen with the mutants. And they've been doing time courses, temperature profiles, reduction requirements. And so we know it occurs in less than 10 minutes, the reaction increases over time. 37 degrees optimal temperature, as you would expect for a human enzyme. Let's see. It requires DTT. We believe the reducing equivalents are needed for keeping the thiols reduced, not for reducing the heme iron. Because as I said, that we are pretty confident that it is already in the reduced iron plus 2 state. And now, in the last few months, they've been switching, instead of horse heart apocytochrome C, to peptides, because everybody likes to be a reductionist. We're called filthy reductionists at WashU because we reduce everything down to the pure protein, but also we like to get the system down, in this case, down to the peptide. So we had a bunch of peptides made. This is the motif, cis, XX, his. This is the human sequence, but it's very similar to the horse heart. There's only like 3 conserved differences to the horse, from the horse to the human. And so then the alpha helix 1 that is necessary is right here, okay? And so the initial is in the black. That's what we start with. HCCS is in the black. And then after 1 hour, we can get this peptide to have covalent linkage. It's a different wavelength. We're pretty confident, let me see. We're pretty confident that the reason it's 555 is it's not released from the enzyme, that peptide. Okay, so in fact, the complexes that we purify, the HCCS human cytochrome C complexes we purify, have a 554. Something unique about the environment of the complex, right, that sort of shifts the spectra from a 550 to a 554. We don't know if that's because there's 2 His ligands, the His 154 and the His 19, or what the reason is for that. But this mimics then the in vivo data as far as the alpha peak of the reduced form. Oh, let me say one thing, and I didn't put it in here, I probably should have. They have peptides that are nipping at the alpha helix 1, and those do not form the covalent link, as Shallon's JBC paper from last year shows. That alpha helix 1 is necessary to position the first cysteine and to bind to the HCCS. So we've got about 6-7 peptides now that we've tried, and it seems to be very consistent with the in vivo data that we've done in E. coli. And so we're happy with the progress of the in vitro. Got a lot more work to do on this, and so that's where I'm going to end. Heme binding is essential for in vitro activity. It requires a reducing environment. Works better anaerobically, but we're starting to get it to work tweaking some things aerobically. It's always easier for the postdocs or students to do things aerobically than anaerobically. The peptides require anaerobic. The peptides, though, they undergo probably disulfide bond formation in a different rate than the apo horse heart cytochrome C, and so still trying to get that to work aerobically, but if we have to do everything in the anaerobic chamber, we will. So the in vitro requirements mimic the in vivo for this, and just end with one of the sort of approaches, or not approach, but goals that my lab is long-term, it's been, real long-term, is to find inhibitors of these pathways. Okay, I sell to my NIH panel. You always have to sell to your NIH panels. And I have sold it for many, many years that the bacterial systems, in Mycobacterium system 2, in all of the most of the gram negatives, system 1, those pathways are quite different than the human HCCS. And so because of the difference, and their substrate recognition is different. I already mentioned cis, XX, cis, his is the recognized motif of the bacterial pathways. It should be feasible to find inhibitors. Everybody sells their stuff in some way. So we sell it that way, that it should be feasible to find inhibitors of system 1 and 2 that don't work on HCCS, or inhibitors of HCCS that don't work on the bacterial pathways, or so on. And so that's where I'm going to leave you. I've already told you Shalyn and Molly, Deanna were instrumental in most of the work that was shown here. A couple of collaborators besides Kenton Rogers. LeVue has done EPR for us, and John Taylor is a chemist, a great chemist in our chemistry department, who has really thought about the reaction mechanisms of cysteine covalent links, histidine covalent links with H130, and has really drawn up the schemes in our reviews that we've written on this for that chemistry to occur. And then my class I teach now is a senior class called DNA Manipulations. And for the last 5 years, I just show a couple classes here. We always have a set of projects for the class, 12 students, that is built around this stuff. Okay, so the TMD, for example, didn't say anything about the transmembrane domain mutations, cysteine substitutions there. Last fall's class, and we're getting some very interesting data on heme going through the transmembrane domains. And we have some proof that actually heme does go through. That's one of the questions in the field that's still outstanding, is does heme go through CCMC? There's not the classic histidines there, and I think we know why, but so on. One other question that I think is important is, why do we have 3 systems, right? Why don't we just have one system if it works well? And I think it has to do with heme reduction, oxidation in the cell. Each one of these has a different mechanism of keeping heme reduced or reducing heme. The second thing it probably has to do with system 1 versus system 2 is system 1 can use heme at very low levels compared to system 2 in E. coli, and we've published that. And so those organisms that have to have heme at very low levels. Remember, having heme at low levels means you have iron at low levels, right? So that's our guess, and whenever something is evolutionary, it's a guess. Often, right? But that's our best guess on why these 3 systems have evolved. Is maintaining heme in the reduced state is important sometimes. Keep using heme at low levels is important sometimes, and that's why we have them. Okay, thank you. Questions?
So in the mammalian system, you didn't show a transporter that got heme across the membrane.
Yeah.
So what is your prediction?
That's a great question. There are a couple mitochondrial putative porphyrin or heme transporters that have been worked on. Iqbal Hamza has a review on those a couple years ago and so on. One's an ABC transporter, and so they may be the reason. I've argued with Iqbal that there's another possibility, that some organisms flip heme you know, if heme is made in the mitochondria, right, of course, and that some organisms may use their heme flipases, their lipid flipases, right? They flip lipids. Heme is like a phospholipid in a way. It's amphipathic and charged on one side and very hydrophobic on the other. And I think Iqbal actually has a recent review in the last few months on that principle. But anyway, maybe he's agreeing. But anyway, there's either specific transport of heme via these putative transporters, or it's a flipase that, just like with phospholipid flipases, flips it over to the other side, and then you may not need a transmembrane transporter. So one of those. I'm hoping this approach with the cysteine cross-linking might help some of the putative heme transport people in trapping heme there. Yeah. John.
Why does this not work on porphyrin? Like porphyrin 9?
Why wouldn't it recognize a porphyrin? Well, in the case of the CCSBA, there is a definitive requirement for the iron axial ligand of the histidines. For CCMC, there's no histidines there. There is a tyrosine, one in the transmembrane domains for CCMC moving through. So I would guess they don't work on porphyrins just because of recognition problems. I mean, non-metallic porphyrins. And they don't recognize it. It doesn't make sense to recognize it. If the ones I've talked about are dedicated to heme transport. And so probably they're candidates for transporters that are porphyrin and heme. And I think Iqbal, some of his HRGs might be those types too, you know.
And will they work on other metallated tetracyclines?
You mean these other ones? Mm-hmm. Or these, or mine?
Yeah.
Other metal porphyrins are not inserted into cytochrome C, nor do they seem to inhibit the normal cytochrome C biogenesis using heme. So that's the result. We've published that tin. I take that back. There's a tin protoporphyrin. This is a 2007 JBAC-T paper that we have. Tin protoporphyrin inhibits when you have a porin that you put in there that can bring in these other metal. Mm-hmm. Tin protoporphyrin can inhibit the pathways. So they could inhibit because they're competitive inhibitors with the natural heme. So that's the only data we have that suggests maybe. Yeah.
So we can return to your speculation on evolution at the end. When do you think mitochondria developed the simplified HCCS system? Given general prokaryotic origin, you might have expected that they might have something that looks a bit similar to system 1.
Right. There's a lot of mitochondrial, there's quite a few mitochondria, the organisms that have mitochondria, that have system 1 still. All plants have system 1. And that really, to me, that speaks to the requirement to protect the heme in the bacterial pathways from oxidation. Plants go through such high oxidation, low oxidation, oxygen evolution. And so all plants have retained system 1. So there's some protozoa that have retained system 1, paramecium and tetrahymena. And so, yet there are some protozoa, Plasmodium, that have evolved into using system 3, the very simple system. And that's another evolutionary question. It's hard to answer. But again, it may be the environment of the heme rather than the actual need for system 1 or 2 in those cases. And evolutionarily, when did HCCS evolve? We have a review in Tibbs last year that Shallon is the first author on that speculates in one of the, in Tibbs you get to put a box, a special box on the evolution of HCCS. And it goes over, the guy's last name is Smith. He's an evolutionary biologist, and he has specifically addressed HCCS in some of his reports. He's from Cambridge. I forgot his first name. But anyway, we sort of use his timeline But we believe it's a little earlier for some other very specific reasons. But maybe 800 million years ago, HCCS started to evolve. And then the true HCCS came about, I mean, the one we know today, HCCS, maybe 500 to 600 million years ago. And those that still have the system 1 just retained it because of the properties I mentioned. And so those that got the system 3 could get away with that because of this maybe coevolution of an environment that is less oxidizing or something. So that's our speculation in that box in the Tibbs letter.
Are the transporters control transcription or post-transcription?
You mean the bacterial ones?
Yeah.
Yeah, that's a interesting story, and each bacteria is different. So you have some bacteria that have many bacteria have cytochrome C oxidase, especially the alpha proteobacteria that evolved into the mitochondria. And so they have to have their system 1 transcribed under aerobic conditions, and usually they have anaerobic respiration also. So they have to have transcription of those genes expressed all the time, basically. But there's some organisms, like E. coli, that their CCM operon of 8 genes is only expressed under anaerobic nitrate conditions. And there's regulators that, of course, sense that and do that. So it just depends on the particular bacteria, on what is transcribed when. Yeah.
All right, thanks, Bob.